They didn't give you they didn't give you.
So welcome everybody to this talk. Our speaker for the talk today is Andrei Manolescu, who is visiting us from Iceland University. He visited us already in 2012. I think it's your second time with us. Well, Andrei did his PhD in the Institute of Atomic Physics in Bucharest, and then he was a guest scientist in different research centers in the University of Reykjavik, Duke University, at the MPI, Stuttgart, ICTP in Trieste, and from 2008 he is full professor in Iceland University in Reykjavik. He is a well-known condensed matter physicist. He is going to give us a IFISC seminar next week. But since he has also worked in other topics like genetics, he's going to tell us today his experience in genetics, his journey to the center of genetics. So, thank you very much.
Thank you. Thank you for the introduction. And I'm trying to repeat a presentation that I had a couple of years ago in Decode in Ankara, where it was a colloquium for students, and my intention, because they were physics students, my intention and my message was that if you study physics, you can do many things. So I was encouraging the students to look into fields which are not traditional physics, but methodology and conceptual concepts and other things are, and I hope you will agree with me, they are quite common to our way of thinking. So the long title is complex title, because this is also the complexity of the field. So there is a small catch in the title. In Iceland, we are in Reykjavik, in this place, and across the sea, 130 kilometers away, there is a mountain which is famous for the story of Journey to the Center of the World. So this is a place where it's a volcano, and it's also an iconic mountain for Iceland. It's a small ice cap, unfortunately it's shrinking on the top, and you can see when a sunny day over this distance, and you see here the typical summer picture, because the sunset is in this direction, is almost to the north, whereas here is to the west. And on the other hand, the catch is that the company where I was working in the Decode Genetics was, and it's still really, the center of genetics, of human genetics, because most of them, it's a major player. And another aspect of my experience is that I was working in a company which in fact behaved like a research institute. Publications, seminars, and the business part was always something that I would say was more of a marginal aspect, or in connection with funding sources, pharma companies, but the pharma companies would benefit of the scientific result. Okay, so the company I was working in between 1999 and 2008 is called Decode Genetics, and you could see it as a national project in Iceland. It has started in 1996 by Dr. Kári Stefánsson, who was a professor of neurology, and the idea was to use the Icelandic population to discover genetic variants genetic forms which are responsible for common diseases. The older idea is that, let's say, a disease or a special trait, say blue hair, if that exists, or red hair, or that can be determined by genes. But if some characters of this type of traits can be determined by a single gene, typically they are rare. These are called Mendelian forms or Mendelian traits. Say single particle states, like in physics. But having already several examples of these kinds of situations, for example, there is a disease where a child is getting old in 3 years. It's called progeria. There is a gene specific to that disease, and if you have that form, you have the disease. But that is usually a rare case. Now, given that some situations like this were solved, so the gene was found through studies on small families, sometimes on twins, then the next step was to try to solve common diseases. Common diseases means diseases that are actually very, very familiar. Cardiovascular diseases, cancer diseases, and then in the world of genetics it was a kind of movement towards trying to use those methods in these complex phenotypes. The phenotype is the character or the way you look or the disease itself. Because Iceland has a relatively small population, but relatively isolated, so it was like in very good configuration to have a large statistical sample size, but also a kind of clean pool, because mixture with other populations would actually create even more diversity. And in this case wasn't a tempting idea. So then Dr. Stefansson was able to convince the Icelandic authorities, and then he got money from Roche, the big pharma company, and established this Icelandic company. And with the goal of exploring the complex diseases. Now, I would briefly show some ideas of the molecular genetics. I'm sure you know, I don't know how much, but just briefly. So we basically have this key molecule, or life is determined by this key molecule called DNA. Observe it is twist. There is DNA left-handed, which is not relevant for life. There is also DNA triple-stranded. So these are more unstable forms. And then we have, in each layer, we have a pair of bases paired in a specific way. The guanine with cytosine and the adenine with thymine. And you see they are connected by hydrogen bonds, which are relatively weak. In the vertical direction, they are connected by covalent bonds, which are strong. So this DNA can split, and then, because of the pairing, on each strand, there will be complementary molecules attached, and then it multiplies. But the amazing thing is that it's highly packed. How it is packed, it's a mystery. If you count, you have 3 billion base pairs in the human genome, from one parent and from another parent. if you think by length, this corresponds to approximately 1 meter of DNA that you inherit from each parent. You have 2 chromosomes, one from the father, one from the mother. Now, the chromosomes themselves are not obviously the linear DNA. The DNA is split into segments, let's say this big or variable, and then they pack, and they form chromosomes. Now, the packing is a mystery. Imagine you take a simple, I don't have, but if you take a simple thread and start twisting, twist, 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 and at some point it's so densely twisted that all of a sudden one branch will emerge and it will twist further. And if you do it, then I would say it's not exactly like that, but this complicated twist actually allows the coiling at different levels. So the first level is around some proteins which are called histones, and then you get something which is a new stretch, which is called or doesn't even matter. It's again, the twist goes again and again in 6, 7 stages until you have the chromosome. So we have 46 in pairs. They are counted approximately by size. So this is chromosome 1, maternal, paternal. Chromosome 2, maternal, paternal. Sometimes when you see them shown in this form, it is shown how do they replicate, how do they multiply. So this part is the centromere. And then you have a approximately smaller length or segment, approximately larger. This is called the P, this is called the Q. P is from petite. Now, the smallest chromosomes are the 21. The particular X chromosome is what determines the sex. If you have 2 copies of X, you always have 2 copies of 1, 2 copies of 2, and so on. If you have 2 copies of the X, then you are a female. If you have an X and the so-called Y chromosome, you are a male. Interestingly, the Y chromosome is very small. And contrary to the legend, it seems that it has been emerged from an X chromosome. So in a way, if at some point in history the organism did not have sex, then little by little one small defect was created as an additional chromosome, and that's how man was coming into nature. So it's more like Adam has been created by Eve and not Eve from the rib of course, these are complicated things, and we can only have some sort of very simplified model, like maybe even more primitive than the Big Bang theory. This is amazing because we know, or at least I would say I would know more about the evolution of the universe rather than the evolution of the life. And how the DNA coils. This is a subject for physics, which is, I would say, in the childhood. One primitive idea is from Boris Shklovsky, who is known in the mesoscopic physics and in condensed matter and semiconductors. And he says that not only he, but people say there must be electrostatics. At that level, there is no other fundamental force. But how complex can electrostatics be? And then he comes with an idea that if you have electrically charged spheres, which are proteins, and basically they are negative because they are lipids, and then he also thinks that there are ions around the molecules. So he has this model that you can actually have the DNA is actually coming dressed with positive ions because the DNA itself is also negative, and then they can create this complex electrostatic equilibrium, and then you can have a certain distance between the histones, and then everything coils again and so on. So again, this is more like a very primitive idea, and I don't want to insist. Of course, this can be other aspects, but again, this is an unsolved problem. Now, I have already suggested that the DNA has a three-dimensional structure, and as we know, that is probably the relevant structure in many situations. But the way biologists think and the way the genetics is formulated is more according to what we can call the linear model of the DNA. So imagine now you stretch the DNA of a chromosome. Chromosomes are independent. We look at one chromosome at a time, but we are not thinking about the three-dimensional structure. We only think about the composition. And of course, we are not thinking about the double-stranding aspect, because if you would do, then you would call each base base pair. But we think in one strand. The information is complementary from the other strand. So suppose you have from the mother a segment of a particular chromosome which has this composition, this sequence. The homologue, the same chromosome, but from the father, the same as number, would be, for example, of this type. What is the difference? You have one letter T here, and from the paternal you have a C. You possibly can have another T here and a G here. But observe, the rest is the same. So the important aspect, and also difficult to understand, approximately 1% or less of the human genome is polymorphic. Polymorphic means that you have two possibilities. The rest of 99% or more is the same. So approximately we differ from each other at this level by a 1% amount of information. When you have a T here and a C here, we say you have two alleles. Allele T, allele C. And when we look at this combination at this particular location, we say this is the genotype that you have. So the genotype consists of two elements. At a particular location, what you have from the one parent and what you have from another parent. So to characterize the genotype, we have this kind of vector. So Here I use notations AB. A more traditional notation in genetics is big A, small a. But now, if we look at one particular location, we have two values, B, two letters, A and T, or G and C, or not necessarily paired, because a pairing is across the double stranding. But now we said we take only one strand and one strand. So there is something called positive strand from each helix. So on another locus or location, you have another two elements. If now you are able to say A1 comes from the mother and A2 comes from the mother also, then you have the so-called haplotype. So you know that A1 and A2 are on the same chromosome. This is a major aspect because in the genotyping, in the way the data is collected, you cannot distinguish between the maternal and paternal. The technology does that, puts all chromosomes together, and through some methods of multiplication of local sequencing sequences, then what the result is a pair. So what you get experimentally is this. If you only look at one particular location, you don't have any problem. You say this is maternal, this is paternal. But if you have more locations, and this is always the case, then you have a problem to say whether A1 is paired on the same chromosome with B2 and the other will B1 with A2, or if they split in this form. So if you can do that, and this is typically done by some statistical calculations, usually the method is called expectation maximization, which is not necessarily used only for this particular problem, then you have the haplotypes. Now, what do we observe in the genome? If we observe something, we say we have a marker. It's like we say in physics, we know an observable. It's not a quantity. It's basically a composition. But that can be one letter. And if that is one letter, like I was showing earlier, just one letter variation, this would be called single-nucleotide polymorphism, SNP. And these are the most used markers now. Just a single letter. The same would be here. We have a SNP TC, so you have a T from the mother and C from the father, or the other way. Or another SNP TG. But more complex markers were more popular in the past, where actually the SNP genotyping was not so easy to produce, because going to one basis is a bit more difficult technically. Now, other types of markers in the genome would be repetitions of the same sequence. So, for example, here, I may have on a particular location, on a particular chromosome, this sequence from a parent, and from the other parent, the same sequence but repeated a different number of times. So here is 1, 2, 3, 4 repetitions. There can be another version, 6 or 5 or 3. Other possibilities is to observe little parts which are deleted from one chromosome or inserted on another chromosome. And of course, the ultimate marker, so to speak, would be the complete sequence. That means 3 billion bases times 2. Now, another key aspect of the propagation of the DNA from parents to offsprings and between generations is the phenomenon of recombination. if you only look at a single point, at a single location, this is the simple scheme. So suppose the male or the father. So conventionally, the square shows the male and the circle shows the female. So these are two parents and they have three offspring. So you have at the father side alleles A and B, whatever they mean. They can be SNPs, they can be microsatellites, they can be anything, abstract. The female has A and C. So now you observe, in this case, the boy inherited the A from the father and the A from the mother. You don't know which one is. They are identical at the particular locus. I'm not talking about the whole chromosome. This girl inherited the A from the father and the C from the mother. And here we have B and A. Now, what happens with the entire chromosome? How it is passed? Well, this is another mystery and a lot of science involved. Recombination phenomenon says that if the blue and the green, they are the chromosomes of the father, red and black, what the father transfers is kind of combination of his two chromosomes. These two chromosomes are going to meet together. There will be a cut in both, and they are going to exchange the material. So the boy receives a recombination of the paternal chromosomes, but it happens that he received the complete entire chromosome from the mother. In this example, it's the other way. Here, both chromosomes are recombined. Recombination occurs approximately once per chromosome on the average. The mystery of the recombination is common with a class of mysteries, recognition. How it is possible that two chromosomes, which are, let's say, both chromosome 1, we could understand why chromosome 1 and chromosome 2, maybe they don't approach each other. Although, I don't know. But fine. Chromosome 1 and chromosome 1, they have to approach each other. And there will be a point where they will merge. They will meet. At that point, there will be a cut and they exchange I can understand they may exchange or may not. I can understand the cut, sort of temperature, things, effects, instabilities. But how do they meet in such a way that they meet at the same analog point? Because if they will meet with this chromosome here and this chromosome here, you do not actually create chromosome 1. So either you lose gene or maybe they meet randomly and only what is transferred to the offspring is the correct mixture. It's difficult to believe that also. But this is also, as I said, another big, and it is not a big mystery. It's not only this one, the big mystery. How proteins can actually, because then this is not happening for isolated chromosomes. There are a lot of ions around. There are some proteins coming and contributing. This is a physics mechanism based on electrostatics proposed by Kornishev and Laikin. I'm not very much aware of, I read something about later on that they could observe experimentally something, but you see, this is a complicated physics. What they say is that because of the electrostatic charge which is distributed on the surface, which comes first from electrons that are situated on the sides, because the DNA is acid, it's negatively charged, and because of the counter ions which are in the solution, which condense and create a complicated charge profile, but charge and charge profile will actually see each other at points where you have similar density, similar distributions. And the distribution is variable because of the twist angles. So the angles of the bases may actually fluctuate. Okay, again, this is only to create, to make even more, interest, not that I was doing that.
So you mentioned about the base pairing, the G, C, and AT. So
Yes.
it's energetically more favorable, or?
It seems so, but on the other hand, energetically, you have to keep in mind that the biological DNA is always in a solution. It's never in vacuum. So it is not clear that you take a chromosome, put it in vacuum, and it will stay like that. It might actually expand.
So I was also curious about the chromosome, which you took as an example. That I saw many mismatches between, but I thought just pairing, you said, G, C, and AT, that's usually, that's most part of the DNA, and other things are very rare, but it's not the case. And I didn't know.
I'm not sure I understand, but be careful. If you think about the DNA, I'm talking about two levels of sequencing, of sequences. This is one simple DNA molecule. It always has base pairs. So A, T, G, C, CG, this is not polymorphism.
So there was not
This is the internal structure. Polymorphism is when you have the next one, and let's say you have T, A, G, C, and possibly T, A. So at this location, now let's put it this way. If we only look at this row and this row, this is what I have. See, now I can. Yes. This is what I have in this picture. So the model reduced basically to the sufficient information and removing every redundancy. Now, the International HapMap project was an intention to observe the haplotypes occurring in the humans. Before that, it was a sequencing project. Sometime in 2002, the company Celera finalized the first sequencing of the human genome. They sequenced 3 individuals, meaning that they collected the information about all letters, so to speak. Before that, that was done on portions of the genome. Now, with the Celera project, sequencing project, it was observed that in fact the genes are even less than people expected. What are the genes? Along the chromosomes, you have small intervals between 100 and sometimes maybe 1,000,000 bases, which would be a large gene, which create, through complicated mechanisms, they create proteins. These are the units that, in fact, create or maintain the life. Or that is what is supposed. In total, genes are not more than 5% of the genome. Now, after having the sequence, then the question was, given the fact that so large intervals are basically identical, can we observe strictly the polymorphic DNA, and then can we observe basically the DNA, I mean, can we reduce the long DNA information to precisely what is variable from individuals? And that was the HapMap project, haplotype map. So they measured haplotypes in 3 populations. Africans, Europeans, and a combination of Chinese and Japanese. These are believed to be the basic populations that can be, so through various linear combinations, we are actually coming from these 3 groups. What has been observed is in fact a truth that was expected, that the variability is not so big. So, for example, if you have a haplotype T, A, G, A, C, let's say in a region, if you look at these 5 SNPs, they can vary from individual to individual, but you would expect 5 to the 4 combinations. In fact, you have a lot less. You could have, for example, A, G, suppose T, C, and T, A, I'm simply picking T, A, T, so let's say A, and maybe a couple of more. So this means that the variability of the segments of those polymorphism is not complete. That is first observation. The second observation is that if you look at those SNPs that are variable, so we can have a lot of bases here, that are identical. So I didn't put them, but then there's another lot of bases which are identical. We are only looking at what is variable. Now, what is observed is that these kind of haplotypes break at some point, and then you have another group, and another group, and another group. So the variable DNA is split into blocks, which in fact is consistent with the idea of the recombination. So what you inherit from, say, Eve and Adam is the same DNA which from time to time recombines and is transmitted to the next generations, and what we get is in fact segments. So the segments are called blocks, linkage disequilibrium blocks. Then I have to comment on this complicated name. Linkage for us, or generally, means that 2 things are linked, correlated. In genetics, linkage equilibrium will tell you that they are not linked. They are independent. So it cancels the linkage. Linkage equilibrium. Linkage disequilibrium will mean they are linked. So this is the evolution of the concept. In fact, it is even said that disease is linked to a specific gene, or it's linkage equilibrium with that gene, meaning that they are independent. But anyway, linkage disequilibrium means correlation. So the genome is split into blocks, and these points, now what I have here is a correlation coefficient between between a number of SNPs, so these are letters, placed in the position in the physical order on the genome. Position 1, 2, 3, and the position goes in this direction. Here I have the same list. When the correlation coefficient is high, then you have a red point or a blue point. These are two different measures. This is a so-called R square, this is a so-called D prime, but that doesn't matter. Obviously, on the diagonal, you have correlation 1 because it's one SNP correlated to the same SNP. But here, you see the block structure, so correlation coefficients, and what you have in these plots is only from the diagonal. So basically, it's a correlation coefficient between pairs of such bases, letters. Now, the disease projects want to find a specific location on the genome which is sort of associated to the disease. How that can be assessed? There are several ideas. So one initial idea was that you would look in families. And in families, there is common genetic material. But if you look in families, in many families where you have people carrying the same disease, let's say myocardial infarction, you would expect that people like that, they would share genetic material in excess relatively to the expectation. Because they would say, you would say, okay, the idea is they would inherit the same gene, if it's one, and then they would actually be more similar than, let's say, other people in the same condition. So evaluating this excess of sharing was done with a relatively complex model, a hidden Markov model, which would take into account the recombinations, the configuration of the family. A family would have 30 to even 50 members, depending on the possibilities of computational possibilities. And then the idea was to try to isolate a piece of DNA through recombinations and see which one is the common to the people carrying a particular disease. Here I have a scheme with blue eyes and brown eyes. So if these people who have blue eyes, they have in common something, ignore the fact that the other ones are brown, then you would actually pick this excess and find that the gene has to be in that region. But the region could be almost a quarter of the chromosome. I worked on hypertension, and we had, with this type of analysis, all chromosomes analyzed, and we find on the chromosome 18 a very peak peak, which corresponds to the so-called lot score, so a kind of statistical measure, which says out of 490 families, we find that this piece of chromosome, which is pretty large, 10 megabases more, would actually contain hidden a variant which possibly would be just a single letter, possibly a mutation, because this is what biologists are after. If you have a letter change in an exon of a gene, which is the key element there, then you have a wrong amino acid. So this is a standard model for them. And if we can find that, it means that we can explain this peak. This peak has large resolution. So to do that, we would put more markers, and in that time this was microsatellites. And then more markers, so if we put more markers, the so-called statistical signal grows. And then we start analyzing in detail the region, hoping to find that excess, which we didn't find. If this is a real result, which nobody knows, it might actually be that there might be still a hidden genetic variant, but we couldn't find it. I mean, okay, so this might also be a statistical power problem. I wouldn't like to call it false positive, but if somebody doesn't like this result, that is what would be called. So, next, we had more possibilities. We had more genotyping possibilities. In another example, we analyzed stroke, another large number of families, and we have some sort of indication, like I was showing before, with the recombination method, but then we find a gene which has some implications known in the biology of vessels, and then we have a lot of data measured on that location, and we find some variant with kind of a effect, relative risk of 2. What means relative risk of 2? What is the probability to have a stroke if you have that gene divided by the probability of having stroke if you don't have that gene? This is the relative risk, and that is how typically these results are characterized. For example, what is the relative risk of having a myocardial infarction if you smoke? Or what is the risk of myocardial infarction created by smoking? It's about 2. If you smoke, if you have a myocardial infarction, if the probability, let's put it like this, probability of having a myocardial infarction if you smoke divided by probability to have and not being a smoker is about 2. The same is a male versus female. So there is a relative risk of 2 for males to have myocardial infarction. Here we have a relative risk of approximately 2 or less. And this is discovered in the Icelandic population. Later, we had data from other populations, and we wanted to replicate this result. It was not replicated. Some aspects, more like a kind of subphenotype with a small tendency. So again, we put a lot of data, we had a lot of statistical evidence, but still this is not replicated. This can be a population-dependent situation or other things. Then, sometime in the middle of 2000, sometimes 2005, when the HapMap project was finalized, or at least the first phase, then we started doing genome-wide scans with SNPs, with single letters. So we could put initially 300,000, then 500,000 SNPs, identify and look for similarity between individuals who had a disease. Similarities, now they are more simply calculated. You look at how many patients have this allele, how many people in the population, and what would you expect if they would be both the same type. So this can be done with what people call Fisher test or chi-square test, and these are simple statistical tests. And we found in myocardial infarction, we found a result on many populations, at least 3. And then we could replicate it. And this paper was published together, side by side, with another paper observing the same thing from a Canadian group. actually I wanted to say about this paper. This result is without a doubt real, but it took a while to be able to cover the whole genome. Now, what is the relative risk of this variant found on chromosome 9, which happens to be in a gene or close to a gene which is known for cancer? 1.25, somewhere between 1.2 and 1.3. So it's a weak effect. This is a typical situation. So trying to find this kind of genetic reasons for common diseases, we end up by finding a weak effect which does not explain the disease. Another interesting case was with myocardial infarction in African Americans. Again, by searching for particular genes and combining information about genotypes and the behavior of the gene, which is in particular related to inflammation, which is a mechanism of the infarction, because apparently it's not only the cholesterol which creates plaques and obstructs the coronaries. It's also the coronary which reacts by inflammation. So then when it expels, then you get some clog in the vessels. So what we find is that this particular gene has a haplotype which is like 10% present in Europeans, Icelanders or European Americans, with a very small relative risk, 1.1, 1.2, but it is of a much higher risk in African Americans. Because the African Americans are admixed, so they are partly original Africans, partly Europeans or Caucasians, but this particular haplotype does not exist in Africa, which would actually fit to an interpretation that because of the admixture, this particular haplotype coming from the whites is actually creating them a damage. This was also a result which was not, I mean, we could replicate it within a small group of populations. I'm not aware of more discoveries, but this was an initial intention to create a drug, which in fact did not go too far. So this is a more interesting aspect from the admixture of populations aspect. And another example, prostate cancer. We could find a place in the genome where there are a cluster of single-nucleotide polymorphisms which are associated with the disease. But there is no gene there. The closest gene is somewhere at a certain distance, and if you see here, there are many blocks between the gene and the location where we are looking at. Which simply means these two pieces are independent. If there is no correlation between them, how can we understand that? How can we think that the gene will be involved? Still, biologists take that gene and say, this is the closest. My interpretation is DNA is 3D.
What is the distance?
300 kilobases. Yeah, but this was, apparently it's not megabases, but everything we did statistically did not show anything. Sequencing, everything. So this is, to me, without a doubt, the gene is not involved. But then the question is, what this part of DNA does, which is not characterized biologically. And the myocardial infarction gene, which happens to be close to or somewhere in the boundary of a cancer gene, tells you that the cancer gene is involved, or simply it's all more or less that. But so what is not, or maybe biologists would say that is guaranteed. But to me, that is, I don't know. The question would be more that whether that gene is involved in cancer rather than when that location is involved in myocardial infarction, because that has been reproduced many times. And finally, an example about smoking. Smoking gene. So if you have a particular form in a gene which has a funny name, CRINA5, in fact it's CRINA3, it's a cluster of gene on chromosome 15, then you would be addicted to smoking. It was a long struggle to define a correct phenotype for smoking because there's so much variability and people have so complicated behavior so in the end we could define a smoking measure, and then we had almost 16,000 people to analyze. And yes, we found the smoking gene. If you have the allele I forgot which one is the risk allele, but there are G and T for this particular location. If you have the risk allele T, then not you, because this is a key aspect. It's difficult to put it on individuals. But on the average, the number of cigarettes per day were larger by 1. So the association is very modest, but the miracle is that we could see with the sample size and with the analysis and everything. But this is also reproduced. So what I want to say is that there are plenty of genetic results about risk to common diseases. Typically they are weak signals, they are small. So this is something that in general you don't get commented too much. When they write in the newspapers, like they wrote about us in New York Times, they found the smoking gene, they found what is the effect? So basically the gene itself does not explain the phenotype, does not explain the smoking or the infarction. But then there are genome-wide diagnostic tests. So I found that the price increased from $99, which I had on the previous slides, to $149. Probably they increased the number of SNPs. You can send them a biologic sample, a swab, saliva, which is easy to collect. At that time we would take blood. And then they can tell you whether you have risk for various diseases, whether you are related possibly to some of these people. But I wouldn't say the information is not unuseful, because it starts now, then let's say there is, or the transition after I left the field was more and more to do sequencing. And now by doing sequencing, the sequencing of the full genome from the Celera times dropped several orders of magnitude in price. And I think this is 2011. So it reaches a couple of thousand of euro per individual. This means that they can solve rare diseases. Diseases that have been observed on few people, which is an example here, they could actually be analyzed at the sequencing level, and there could be a gene identified. So now you can identify Mendelian traits which are highly predictable. They are rare, but they are deterministic. And that is the advance that is now. So one idea is then, of course, it's a lot of ethics in the field. One idea is that ethics is, of course, to treat everything confidential. But if you know someone is exposed to a disease and not with a relative risk 1.5, this is nothing. With 6, then would it be ethical to try to make to do something and inform? Of course, within some ethical limits and some other. So this is what is going on. And then diagnostic tests, if you possibly have many of those variants which create low risk, but put together they increase your risk, so then it's also valid and valuable information. So, kind of conclusion, I would mention that there are common effects which are usually weak, and rare effects which possibly they sit in families, and that's why actually they are family specific. So this is an example which is the relative risk of a genetic variant in obesity might be high if that is a special type of obesity which needs to be characterized. But in general, maybe it's more important what you eat. For rare cases, then in families where these rare variants exist, they are not rare. They are rare across the whole population. But with this sequencing methodology and this present stage of genetics, they can possibly be identified. So I wanted to finish earlier, but sorry, I even skipped a number of slides.
Thank you for this excursion on genetics, the journey on genetics. I don't know if there are questions or comments.
Maybe a very simple question. You demonstrated in one of your slides that there is some correlation which was observed when you analyzed different genes in United States, European countries, Asia, and African countries. And this is the most simple example where you can see how one or another gene dominate in one of the another nation. Did you get this result from this correlation? It was a little bit vague. I mean, this is the most simple, which can give us some information, because blue eyes, and this is a little bit more complicated. No, earlier. One of the first of you. Yes, this is it. I mean, you talked about different nations and...
So if the simple answer, or you talk about this block structure of the DNA. Yes, the block structure is different from the Africans, between Africans, Asians, and Europeans. And indeed, the typical situation is that when the blocks in regions of the genomes are smaller in Africans, because further evolution occurred there. They are much older population. And Africa, they were like an initial population that did not evolve that much until now. But related to this, the story with the African Americans and myocardial infarction has to do with that, because in this gene where we had the haplotype, which was with very modest effects in Europeans, but very strong effects in African Americans, if you look at this table, these are all possible haplotypes that are detected. So we have far less than 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 to the 4, 10,000. But here are the frequencies. So you see, on particular populations, some of them they don't exist, but they do exist in other populations. And our haplotype is something that actually did not exist in African Americans. And here, in the middle, in Africans, here in the middle, you see, this is the structure of the block for Africans. This is the structure of the block for Europeans. So you see this. The European block is actually split in Africa. So because of longer evolution, there is an additional recombination in the middle of the block. So in this sense, yes, then the blocks have different structures between these populations.
When you say relative risk, 1.2 is low, for instance, but doesn't it have to be compared to some measure of the relative risk that you can expect overall? Right?
Yes.
So how genetically, I mean, how does it depend on inheritance at all? I mean, I guess there may be diseases that are...
So as a genetic risk, it might be not low, not high. This is what it is. But in the medical world, the idea is what makes you sick of diabetes. For example, you eat too much sugar. Maybe I'm wrong, but this is. Or what makes you, what gives you lung cancer? You smoke. So there are already factors, risk factors, known by the medical community. Genetic risk factors are only particular risk factors, and they have to be compared with this kind of biological risk factors. High cholesterol, you are exposed to infarction. But now the idea is that if you have a genetic risk of this modest value, would it make sense to create a treatment? Because this is the next problem, because this was the whole idea behind. We make treatment, genetic treatment, personalized treatment, African treatment, European treatment, and so on. But if the risk is so low, then there is no medicine without side effects, and then you don't have a correct target. Although we even did patents on genes. I have some patents on the information. Yeah. So, but on the other hand, if the risk is comparable to those known from biology, then there must be some consideration. And in many cases is that, for example, there is no gene of cancer. Although it is the same name, as much as we put together all kinds of combinations of genes we found in prostate, breast cancer, lung cancer, which we did, nothing can be said, because it's more even diluted. So apparently there are. So cancer and many of these diseases, in fact, is the endpoint, the biological effect. What is behind? For the same type of cancer, there could be behind several mechanisms. As I say, some of the rare but strong mechanisms are being identified.
Maybe one last simple question again. So you say that, if I understand correctly, that we are distinguished by ourselves, I mean, different people, because only, say, two registers are different. You have molecular, and you say only two, when you demonstrate the cells, and you say G and T, and when you compare that all people distinguished because of different combination, only of two, they say register, translated from language of computer.
Yeah.
Only this difference. The rest is equal,
Yes.
the same.
Yes. As I said, 1% of the genome, if not 0.5%, is clearly at most the
Does this
value.
distinguish genius from normal people or fail?
Even except chromosome X. Yes, genius from normal people, obese from slim people, yeah. And even between species. I think we are not more different than 80% or maybe 10% or so complement from those genes. The mouse is a very nice model of humans, only that it has some parts of chromosomes transferred to other chromosomes. So it's a little bit of mixture. But that is another mistake. But again, this is only if we believe in this model of deterministic of the DNA, because everything here has to do with the idea if anything happens to you, you have a disease, that is because, not because you eat too much ice cream, because you have a gene which tells you to eat, which makes you to eat, not because you are exposed to some environmental aspects, because you have a gene which is, or some genes which are sensitive, so if you move to a cold country, you could be healthy because you have some genes that if you stay in hot, they do something. And this is the model, but in fact, maybe there are many other things happening at other levels. But the geneticist will say, even if it happens at whatever protein level, it is still because there is a gene which does that. This is all based on the idea that you are basically a DNA, a sequence. But what is the effect of 3D structure? Because if there is an effect statistically, and let's say this is observed and it is accepted, and if it is in the place where there is no gene, for me, the DNA is basically not linear. It's a complicated configuration. And if this piece of DNA is supposed a little bit heavier, but sits on top of a gene which is megabases away, but coiling is just underneath, then there might be some physical reasons that the gene is suffocated a little bit. This is something for the rest of the millennium. So
but we already know that this genetic model that you say is very incomplete. I mean, you have genetic regulation and transition factors and so on, and now all the new world of epigenetics. I mean, the histone modification, methylation, things. In particular, means that I mean, your sons and daughters can inherit traits that are not in the DNA. You know. I mean,
yes. So these are de novo mutations. Yes, so this is also a possibility.
Okay, we can stop here. Let's thank the speaker again.